Gastrointestinal Physiology, exploring the chemical regulators of digestion, a deep dive into the messengers that orchestrate digestion. Welcome to the Everything Surgery podcast, where we dissect the extraordinary. Today, we are taking a closer look at the fascinating world of gastrointestinal physiology, focusing especially on the substances that chemically regulate the digestive tract. Rather than relying on abbreviations or tables, we will walk you through each concept in detail, ensuring a smooth and formal narrative, just as you would expect from a top-tier podcast episode. The chemical control system of the gastrointestinal tract. Beyond the anatomical structure and intricate nerve pathways, the digestive system features an elegant chemical control system. This system comprises hormones, substances that act locally, and neurotransmitters, all working together to guarantee that each segment of the digestive tract responds optimally to food intake. These chemical messengers fine-tune motility, secretion, and absorption, making sure resources are used efficiently and every stage of digestion is properly synchronized. Categories of gastrointestinal regulatory substances. Hormones are released by specialized cells into the bloodstream, traveling throughout the body to influence distant target organs. Paracrines are secreted and act locally on neighboring cells, never entering the circulatory system. Neurotransmitters are released from nerve endings within the digestive tract, facilitating communication between nerves and other cells. There are also substances secreted directly into the digestive tract's interior, acting on the lining and contents as digestion proceeds. The 5 principal gastrointestinal hormones. Gastrin. Let us begin with gastrin, a hormone produced by specialized cells in the lower part of the stomach and the upper segment of the small intestine. Gastrin is released in response to the presence of peptides and amino acids in the stomach after a meal, as well as by the stretching of the stomach walls. Neural signals from the vagus nerve, mediated by a specific peptide, also promote gastrin release. Gastrin's principal roles include stimulating the secretion of gastric acid from the cells lining the stomach, promoting the growth of the stomach lining, and enhancing stomach movement. Its secretion is tightly regulated, and when the stomach's acidity drops below a certain threshold, another substance called somatostatin inhibits further gastrin release. Cholecystokinin. The next key hormone is cholecystokinin, produced by cells in the upper portions of the small intestine. Cholecystokinin is released primarily when fatty acids and certain breakdown products of proteins enter the intestine. Its main actions are to trigger the contraction of the gallbladder, promoting the release of bile, and to relax the muscle that controls the exit of bile and pancreatic juices into the intestine. Cholecystokinin also stimulates the pancreas to secrete digestive enzymes, slows the emptying of the stomach to allow for adequate digestion, and signals feelings of fullness to the brain. Additionally, it supports the growth and function of the pancreas. Secretin. Secretin is synthesized by cells in the upper small intestine and is released when acidic contents enter from the stomach. Its primary function is to signal the pancreas to release bicarbonate, a substance that neutralizes stomach acid, protecting the delicate lining of the intestine. Secretin also encourages the ducts of the liver and pancreas to secrete bicarbonate-rich fluid, and works alongside cholecystokinin to optimize digestive enzyme delivery. Importantly, secretin suppresses the further release of stomach acid, helping maintain a balanced environment for digestion. Glucose-dependent insulinotropic peptide. Glucose-dependent insulinotropic peptide is produced by cells in the upper small intestine and is released in response to glucose and fats entering the digestive tract. Its primary role is to stimulate the pancreas to release insulin, helping the body prepare for the absorption and utilization of nutrients. While it was once known for its ability to inhibit gastric acid secretion, this effect is now considered secondary compared to its role in promoting insulin release. Motilin. Motilin is generated during fasting periods by cells in the upper small intestine. It plays a unique role in stimulating rhythmic contractions throughout the digestive tract, a process known as the migrating motor complex. These contractions help clear out residual food particles and bacteria between meals, maintaining gut health and preventing bacterial overgrowth. Key local messengers. The paracrine somatostatin. Somatostatin is produced by specialized cells located throughout the digestive tract, including the stomach, pancreas, and intestine, released in response to acid and other stimuli. Somatostatin acts as a universal inhibitor. It suppresses the release of virtually all digestive hormones, reduces gastric acid secretion, and limits the secretion of pancreatic and bile fluids, thereby putting the brakes on digestive activity when necessary. Histamine. Another crucial paracrine is histamine, which is released from cells in the stomach lining in response to stimuli such as gastrin and nerve signals. Histamine acts locally to powerfully stimulate acid secretion by the stomach's parietal cells. This mechanism is an important target in the treatment of ulcers, with specific medications designed to block histamine's action and thereby reduce acid production. The integrated phases of digestive control. The regulatory substances we have discussed work in harmony during the three main phases of digestion. In the cephalic phase, the anticipation of food, its sight, smell, and taste, triggers neural signals that prepare the stomach for incoming food by stimulating acid and digestive enzyme release. During the gastric phase, the arrival of food in the stomach further stimulates acid production and enzyme release through both mechanical stretching and chemical signals. Finally, in the intestinal phase, the movement of partially digested food into the small intestine prompts the release of hormones such as secretin and cholecystokinin, which slow the emptying of the stomach and stimulate the secretion of enzymes and bile needed to complete digestion. Conclusion. In summary, the gastrointestinal system is orchestrated by a remarkable network of chemical messengers. These substances coordinate secretions, movements, and absorption processes, ensuring that the digestive tract adapts seamlessly to the volume and composition of each meal. The interplay among hormones, local messengers, and neural signals underpins the efficiency, adaptability, and precision of digestion. Thank you for joining us in this episode. If you enjoyed our content, please remember to like, share, and subscribe. This has been the Everything Surgery Podcast, where we dissect the extraordinary.